All right, let's finish up chapter 6. We've got two more stories to go by. We're sort of going back in time. I moved these out of the way because I wanted to make sure we talked about chapter 11 in person, at least a little bit to get started. So these are things about what you do with the enzyme kinetic parameters, and these are different kinds of thinking that you can do. This will help you on homework problems. And so here's one thing you can do with chymotrypsin. You can give it proteases, you can give it small molecules that look kind of like a peptide that it cleaves. Most importantly for chymotrypsin, that binds ringed molecules, right? So you give it a ringed molecule, and then you put an amide bond for it to cleave, like right next to it. And so you want to see how these things work, and you want to see how the residue next to the ringed molecule has an effect. This is actually, so you see the amide bond, I don't know if you can see my thing, but in substrate A, the amide bond is on the left. Then the ringed molecule is in the middle, and so you see that then it extends past it. So the question is, we know that P1, the ringed molecule, is really important. Does P2, the next residue, does that have any effect on how things work? And that's what this is testing. This is going to test to see if it affects the catalysis of breaking the amide bond or the Km of the substrate binding to the pocket. And so substrate B, you've just extended the peptide a little bit more, and you see that that actually will lower the Km. It binds a little bit better, and the Kcat improves a little bit. So the Kcat Km improves a little bit, so it actually helps to cleave the amide bond a little bit if you add this backbone sort of thing. This is like if you had a glycine on the other side of the phenylalanine. What if instead of a glycine you had an alanine? So you put in the next molecule, which has just a little change. It has basically a CH3 where it would be if you had an alanine next to that phenylalanine. And what's interesting is that the Km is actually not changed. If anything, the Km gets a little worse, but the Kcat gets a lot better. So something about having just that methyl group somehow helps the chymotrypsin bind to this substrate a little bit, but it's more about positioning the amide bond so that it gets properly cleaved. Overall, the Kcat Km goes up by a large factor, by a factor from 2 to about 114. And so we can talk about dividing these two numbers to say how much does it go up. You know, it goes up 5 times from substrate A to B. It goes up 10 times and another 10 times from substrate B to C. So it looks like substrate B to C actually is more important for the enzyme working like an enzyme than A to B. You can do things like that. So just be ready for that. You can also do equations. What about changing the P1? Have you ever wondered, phenylalanine is good. How much worse does it get if I mutate that phenylalanine away? And so what they can do is they can do a different substrate, but here you're changing the P1 rather than changing the P2 position. And you can just do the things where you have the side chain as phenylalanine. That's sort of your default control sort of mode. The Kcat Km for having a phenylalanine in that pocket is 1 times 10 to the 5th. But then you can say, I can substitute these smaller, you can sort of melt that down, and you can say, as I get it smaller and smaller, if I bring it all the way to glycine, my Kcat Km goes all the way down to 0.13. If I bring it down to valine, it's 2 in these units compared to 10 to the 5th. So you can say, okay, how important is it that it has a ring? It's pretty important, but it's also important that it just has carbons there. Even if they're not in a ring, norleucine still sort of works. So you just want to be able to compare the numbers and think it through as far as what does it mean for how structure relates to how this chymotrypsin works. The active site of chymotrypsin is, they even knew for a long time that these 3 residues were what were important for chymotrypsin. If you mutate the 3 residues, you end up getting bigger than expected changes in how chymotrypsin works. And what was interesting is they already knew that the binding pocket was located in one place, but these weren't in the binding pocket, so they must be something about how it catalyzed the protein. And so I just want to point out that you know from these numbers that you have aspartate, histidine, and serine. They are dispersed throughout the sequence. So they knew that these must be folded together to be brought to be right next to the substrate. Because when you mess with them, when you mutate them, the substrate doesn't get cleaved anymore, although it might bind. So these probably affect KCAT more than KM. And so just remember that we've actually seen these three residues before. And you want to, as you're studying it, study these three residues, because we showed you even serine 195 was that nucleophile that we had. And remember, and his 57 was the conjugate base that we had that allowed you to do the acid-base catalysis that allowed the covalent catalysis. The other thing that we showed you, so we showed you that, but I want to show you some of the evidence for why we showed you that, how they figured it out. It doesn't involve crystal structures, it involves biochemical reasoning. And then we sort of had a little cliffhanger, what happens after this, and I want to mention that that's important too. So, what is the evidence that we actually are doing covalent catalysis? Remember, covalent bonds are very strong, but they're also slow. They're slow to make and they're slow to break. And in fact, the slowness of the reaction allowed you to see that there was at least 2 steps, and that the covalent bond must be formed. A slow step must be in the middle of things. And they used this. This is actually a molecule that sometimes we use in the lab because it's a really nice color-changing molecule. When you add the nitrophenyl acetate, it will actually bind to the molecule, and it'll actually be cleaved to paranitrophenol, which notice how that looks like an aromatic residue. So even though it's not perfectly structured, it's kind of the right structure to have an aromatic residue and then to have a cleavable bond next to it. And the greatest thing is it changed color, so you can measure product from just measuring the color of the solution. So what they figured out is that the first step, producing product, would happen quickly at first, but then it would slow down. What you usually expect for something is you expect a smooth curve, maybe a hyperbola, but a straight line at first, right? And you see that straight line at first when you're running this. But then you see it curve down. And so it's not good to use that for an undergraduate lab, but it allowed the scientists to be able to say there's something complex going on here. So if you do chymotrypsin like this, you see that elbow and you say there must be 2 steps. They realized that the 2 steps, they use different concentrations of enzyme and paranitrophenol, and they noticed that in every one of those cases, you got the second slow step seemed to happen. So you have a fast burst phase where you have a lot of color being formed, and then it slows down. And it always slows down right when it gets to the point where you have 1 mole of product for every 1 mole of enzyme. So they were like, oh, that seems like the enzyme is able to do the first step really quickly, but then it gets slowed down. And when you have the enzyme all full, when you have the empty enzyme, it's able to go really fast. But it gets full because it has this slow step. It must be a slow step. It must be second. And it must be a breaking the covalent bond. Therefore, you must make a covalent bond if you have to break it and it's slow. So the fast precedes the slow, 1 substrate per enzyme. This led to the idea there must be a nucleophile. Oh, look, there's that serine. Oh, look, there's a histidine that can pull the proton off and make the serine O- so it can attack really easily. So that's the slow step. And so they have this model that you have the fast step that produces the paranitrophenol. And notice that that's when the bond is actually, the amide bond is broken. Then you have to regenerate the enzyme before you can use it again. And so they developed other things. When the enzyme is regenerated, it produces acetic acid. So I think they did stuff with pH where they measured the acetic acid coming off, and they found out that was the slow step. So eventually there's a bond that gets broken. So how is it broken? And this one thing is, I want you to look at a figure. I think I had the right number for this figure. I want you to look at, there is an achieve question about putting the steps in order. I want you to look at that figure, and then that will have the steps, and basically look back and forth. I'm counting on you having the book open. I want you to be able to interpret the information, put the steps in order, so that you know something about how you deacylate the enzyme. To break this bond, there's a whole another set of enzymatic steps that will complete the cycle of how chymotrypsin works. Okay, and that's all you need to know about chymotrypsin. One more thing you need to know about hexokinase. Hexokinase is an example of something that we mentioned in class. Remember that we talked about induced fit. And we haven't talked about how it's important. It's actually important to how hexokinase works. So hexokinase will catalyze a particular kind of reaction. We had a name for these. We had categories of reactions. We mentioned this before, but I want you to notice I hid it so that you can see it now. It's important. It catalyzes a group transfer reaction where basically you have a glucose and you have an ATP. You take a phosphate group and you transfer it from the ATP to the glucose. This is the first step in glycolysis. Biochem 1, Biochem 2, you're going to start with this when you talk about glycolysis. It's the first thing in Biochem 2, so you're going to be ready for that. But we want to talk about it as an enzyme, because this enzyme actually has a pretty hard job. ATP will be hydrolyzed by this reaction, and you want to hydrolyze the ATP and immediately put that phosphate onto the glucose. But if you don't have glucose around, glucose isn't always constant, right? If you don't have glucose around, the enzyme has a danger that it might hydrolyze the ATP for no reason. And it might put the ATP, the phosphate from the ATP, onto water. In which case, you've just made an ATP burning enzyme that will take all this ATP that you've worked so hard to make. You want it to go to the sodium potassium ATPase. You don't want it to go to you don't want an enzyme that basically just chops up all the ATP and makes it useless. So what you normally do is, okay, we'll do the group transfer reaction. We'll make it work for the pKa. We'll make it work for the different pKa than for a particular pKa, and we'll use acid-base method to do that. You can do that pretty easily. But there's a problem here, because we need to transfer it to a pKa that's an OH pKa that is pretty much the same pKa as water. So we can't do the acid-base method of transferring it. We have to do something else. And that's how it does induced fit. If you see the bound enzyme, you can see that it's kind of bitten down on the sugar molecule shown in red in the middle. The unbound enzyme, with no sugar in the middle, the whole enzyme is floppier. It's opened up. And so the sugar binding site is on the very inside, and the important thing for me to show you, ATP binding site is right here. If you can see my arrow, you can see that the ATP binding site is located between the two parts of the enzyme. That's only there, it's only brought together when the sugar binds first. The sugar has to bind and bring the site together. If there's no sugar, look what happens to the site. It completely falls apart. So the ATP binding site is in two halves, and they are only brought together by the sugar being there in the first place. So this is a case of induced fit, and it's why you might want induced fit, because it allows you to wait until ATP only binds once glucose is bound. You have to wait for the glucose to be there, for the OH of the glucose to be there, before the ATP binds, and then it transfers from the phosphate over to the OH, and you can't have a water being there. One of the ways we show that this works is we use something that has all the binding machinery, but not the catalytic machinery. If you use xylose, and we'll talk about this in just a second, xylose is a sugar that is shorter than glucose. It does not have that OH on it. And in fact, it doesn't have the exact OH that the phosphate is transferred to. So if you use xylose, when xylose is added to hexokinase, then the enzyme will work just fine for binding and hydrolyzing ATP, but it will transfer the phosphate to water because there's no carbon in it where that is. So that's a little bit of a preview. I want you to see that preview. If you're watching this in order, then we will talk immediately about what the structure of xylose is and why it is that I can say that if you don't have that sixth carbon, how can I say that xylose is shorter? Well, that's because of sugar chemistry, which is the next topic right here. So it all fits together, and you can watch that when you are ready for it.